action potential and synaptic transmission. Hmm. The cells in the nervous system communicate with each other and with the rest of the organism using synaptic transmission, meaning communication uh, between two cells is over a very, very tiny space between the two cells, and this requires an electrical discharge. In order to have an electrical discharge, you have to have a potential, uh, and that potential is an electrical potential of the cell membrane. Most cells have a difference in voltage between the inside and the outside. The inside of a neuron, a nerve cell, is usually around 70 millivolts, more negative compared to the outside. Uh, so, how do you get this potential? This is called a resting membrane potential. Um, well, ions cannot pass through the cell membrane because they're charged, they're polar, and the membranes are made of fat, which are uh, hydrophobic, and only neutral things can go through it. So electrically charged ions cannot pass through the membrane. They have to go through channels. And those channels are proteins that are embedded in the cell membrane. And the opening of those channels that will allow ions to go in and out of the cell uh, can be done in several different ways. We can have chemical messengers like hormones or neurotransmitters that, when they bind, open the channel. Mechanical distortion of the membrane can open some types of ion channels. Um, Electrical properties of the membrane. If the membrane voltage changes, that is going to open some other voltage gated channels to allow ions to go in and out. Uh, and then there are so-called leakage channels that open spontaneously. Every now and then they open a little bit, allow a couple of ions in and out, and that way, the ions can leak in and out. In all of those cases, whenever there's an ion channel open, the ions are going to go through these channels following their own concentration gradients, which means if there's more of an ion on one side of the membrane and less on the other, then the ions are going to go from more to less, where there's more to less, in order to equalize the concentration of ions on both sides of the membrane. So, in order to make an unequal distribution, to make sure that there's more on one side than the other, that requires energy. And that energy, like everything else in the body, is provided by the molecule of ATP, that is the universal energy currency in the body. The molecule that does this is a protein embedded in the cell membrane, which is called the sodium potassium pump, or ATPase, uh, and it is an active ion transporter. So what it does is whenever it uses energy from a molecule of ATP, it pumps sodium, 3 ions of sodium, out of the cell, making concentration of the sodium greater on the outside than on the inside, and at the same time moves 2 ions of potassium into the cell, making sure there's a greater concentration of potassium inside of the cell than out of it. Remember that cells also have those leakage channels, so occasionally there's a sodium or a potassium ion going in and out, following their own concentration gradients, not using energy, just going from greater concentration to lower concentration, trying to equalize it. So that ATPase, or potassium pump, has to constantly use energy from ATP to maintain the resting membrane potential, which in most neurons is about minus 70 millivolts, so more negative on the inside than on the outside. So, uh, resting potential is the potential or the voltage across the membrane of a cell that is not in any way stimulated. This is not getting any kind of inputs. But an impulse, a signal coming from another cell, another neuron or sensory cell, can be an electrical impulse, or it can be synaptic communication from another cell, is going to result in opening of some ion channels locally. So let's say on a dendrite, and this will change the membrane potential on that part of the dendrite. And it's usually very small change in voltage. This is called a graded potential, and it's not enough. It's going to start moving down the membrane and dissipate, and it's over. So it's necessary for a neuron to get stimulated that it gets multiple stimuli at the same time. So if you have a whole bunch of stimuli happening simultaneously or almost simultaneously, they add up. So stimulatory inputs are going to depolarize the membrane, making it more positive, and inhibitory are hyperpolarizing impulses. They're going to make it more negative, even further away from zero. So even harder to get it stimulated away. So the neuron is going to do what is called summation. It's going to add up all the stimulatory and all the inhibitory impulses, the pluses and the minuses, adds it up. So if the sum of those impulses is large, it can go further. Instead of just locally changing the membrane potential, the membrane potential is going to trigger the changes in the voltage next to it, and next to it, and next to it. It can go down the dendrite, over the soma, and all the way to axon hillock, the beginning of the axon. Uh, this is called summation. Summation can be spatial, so I have lots of impulses from lots of other cells, and they're all stimulatory, or most of them are stimulatory. They add up because they're happening simultaneously at the same time. Another way to do this is what is called temporal summation. That means the impulse can come from another single cell, but that single cell is going to send a whole series of inputs at a rapid frequency. Pa, 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 pa. It's going to add up. So each one of those impulses, even if it comes from a single cell, is going to increasingly stimulate and stimulate and stimulate and stimulate more and more that local part of the membrane until it gets big enough that as it tries to go down a wave through the membrane of the soma to get all the way to axon hillock. So the axon hillock, if something comes there, it's for the axon hillock to be stimulated, that change in voltage has to cross a threshold. And for most neurons, that threshold is around minus 40 millivolts. So we're starting with minus 70 or so. And then, its stimulation, depolarization, is going to bring it up to minus 40. It's closer to zero now. That's a stimulus that crosses the threshold. What is that going to do to the axon hillock? That opens a sodium channel or two or few at the axon hillocks. What happens? Sodium is going to go down its concentration gradient. It's going to go from where there's a lot of sodium to where there's little, from the outside to the inside. So when you open a couple of these sodium channels at the axon hillock, the sodium is going to rush in down its concentration gradient. That results in a little more depolarization, right? The positive charges are going in, making it more positive on the inside than it was before, which results in opening more sodium channels in the same spot, which depolarizes the membrane even more, which opens even more sodium channels, and more depolarization, more sodium channels. That is a positive feedback loop. At the end, all of the sodium channels are open in that spot, and the membrane potential is now positive. It can be plus 40, plus 50, plus 60 millivolts. So very, very quickly, in milliseconds, you're going from minus 70 to, let's say, plus 50. That's a large difference in voltage, and switching from minus to plus, from negative to positive voltage. So once it gets all the way up to the maximum, all the sodium channels are open, all the sodium is kind of rushing in, and it's maximally depolarized to, let's say, plus 50 or something like that. Then that is going to trigger the opening of the potassium channels. It's the same kind of process. It opens a few that's going to result in potassium, of which there's a lot more inside, to surge out down its concentration gradient from where there's a lot to where there's a little. And that is going to result in repolarization, going back to being polarized, and which is going to result in the opening of more potassium channels. So more potassium can surge out, which repolarizes the membrane even more, which opens more potassium channels, which repolarizes more. It's another positive feedback loop. Once all of the potassium channels are open, by that time the sodium channels are closing or have already closed, and the membrane has now completely repolarized. And it's gone not just all the way down to minus 70, but a little bit more. That is called an overshoot or undershoot. And it takes a little bit of time, there's still micro to milliseconds for ATPase, for the sodium potassium pump, to catch up with it and bring it back to minus 70 again. This whole process, when you start with a resting state, and then the graded potential comes from the dendrites and soma to the axon hillock, reaches a threshold, which results in opening of sodium channels, which results in depolarization of the membrane all the way to its maximum, which results in repolarizing due to potassium channels now being open, the undershoot, and then back to resting state. All of that together is a process called the action potential. So an action potential is generated at the axon hillock at the very beginning of the axon, but the business part of the axon is its end. So that action potential needs to move from the beginning to the end of the axon. And remember, in some nerves, they're a meter long, 3 meters long. If you're a giraffe, they can. It takes time to get there from the beginning to the end. So normally, you would think about it when a cell changes the potential across the membrane at one spot, that is going to move to the next spot. The ions are going to move next door, and that is going to change the cell membrane. You have an action potential in the next portion of the membrane, and the next one, and the next one, and the next one. So that takes time, right? Now, invertebrates, like insects and squid and those kinds of things, the only way they can make that process faster is if they increase the diameter of the axon. So some of them are so thick that you can see them by naked eye, can poke electrodes in that and study them. Invertebrates, like us, there are glial cells called Schwann cells that wrap around the axons and form myelin sheaths. Those myelin sheaths are serving like an isolation tape. If you've ever done any kind of electrical work, you put isolating tape around the wires. So what happens is that action potential that's moving from the hillock next door gets to the portion of the axon where there's myelin sheath. It doesn't have to go gradually through it. It can jump to the other side, to the other node of Ranvier. Remember that one? And jumps to the next one, jumps to the next one, jumps to the next one. So it doesn't have to go gradually from one spot on the membrane to the next one, to the next one, to the next one for another meter or two or three. It can jump. That is called jumping or saltatory transmission of the action potential. So it can get to the end of the axon much, much faster. So what happens when it gets to the end of the axon? So as it's going, everywhere it goes, that action potential is going to trigger opening of sodium channels, depolarization, so opening of potassium channels, repolarization, a little undershoot, and back to the resting. In the meantime, the action potential has moved to the next place, and next place, and next place. At the very end, you don't have sodium and potassium channels anymore. Instead, you have calcium channels. And there's going to be a lot of calcium on the outside. Remember calcium homeostasis. You really have to have a pretty precise concentration of calcium outside of the cells because of this process. Because once the action potential gets to the very end of the axon, it's going to trigger the opening of those calcium channels. Calcium triggers surges in, and that surge in of calcium is going to stimulate a whole bunch of vesicles. Those are small packets that are filled with a neurotransmitter, and each brain cell, each neuron has its own neurotransmitter. It's extremely rare to have two neurotransmitters used by the same cell. So you're going to have a neuron that uses a particular neurotransmitter. And so those vesicles that are made of little membranes that make little spheres, little packets, is the same type of membrane as a cell membrane. So the calcium is going to, through a very complex process, get those vesicles to fuse with the cell membrane to open up and release the neurotransmitter outside of the cell. Where is that outside of the cell? It's in the synaptic cleft, which is that very, very narrow space between the presynaptic cell, which has just finished its action potential and released neurotransmitter, and the postsynaptic cell, the next one. It can be another neuron, it can be a gland, it can be a muscle, like a neuromuscular junction, and that neurotransmitter has very, very little space to go across, which is wonderful because almost instantly it's going to find a neurotransmitter receptor, a very specific receptor for that neurotransmitter. Neurotransmitter binds to the receptor, and receptor is going to react in some way. For instance, it might open a uh, ion channel, which means sodium or something is going to go into the next cell, which is going to change the cell membrane potential of that postsynaptic cell and tells it to do something. It can, it's going to do the summation and all the processing to figure out what to do next. That is synaptic transmission. Uh, so there are neurotransmitters that are stimulatory. They depolarize the membrane, make it more positive, make it easier to sum up to get to the action potential in the next cell. Other neurotransmitters are inhibitory. They hyperpolarize the membrane. They make it more negative, so even harder to produce an action potential in the next cell. So almost all communication between a neuron and the next cell, between a sensory cell, which is a modified neuron, and a regular neuron, or neuron to neuron, or neuron to muscle, or neuron to gland, is done this way. You have an action potential that results in the release of a neurotransmitter in the synaptic cleft, which then in some way informs, or stimulates, or inhibits the next cell, and that is synaptic transmission. There are only a couple of places in the body where nerve cells or modified nerve cells into sensory cells have a different mode of transmission, and that is direct signaling through gap junctions. There are some cells in the retina of the eye that communicate that way because that's much faster. Ions go straight from one cell to the next through a little gap, through a little hole. The other place is the suprachiasmatic nucleus, that is the setting of the pacemaker of the circadian rhythm. Some of those cells communicate via gap junctions, so ions can go straight from one cell to the next. That's much, much faster than synaptic transmission.